Hello and welcome to SpecTransfer. Today we'll be looking at topic 3.2.4 Cell Recognition and the Immune System from the AQA A-Level Biology specification. Because this is a large topic to cover, I've split this topic into two parts, and to watch part 2 of cell recognition and the immune system, just follow the link at the end of this video. So as always, here we have the specification. We'll be starting with an introduction to immunity and what actually leads to the immune system being triggered. This leads us nicely on to antigens, for which we'll need to learn the definition. I'll cover the bit on antigen variability when we talk about vaccines later on. Then we have phagocytosis and how they destroy pathogenic material, followed by the two types of immune response, the cell-mediated and the humoral response. Then we have vaccines, as well as the difference between active and passive immunity, followed by HIV, monoclonal antibodies, and finally the ELISA test. But first, let's start at the beginning of this section of the specification. In the body, we have two types of defense mechanisms. We have specific defense mechanisms and non-specific ones. Non-specific defense mechanisms are immediate. They're the same for all pathogens and include physical barriers such as tiny hairs which trap mucus in your ears, for example, as well as phagocytosis. Specific mechanisms, in contrast, are slower. They are specific to each pathogen and involve lymphocytes. The two types of specific response are the cell-mediated response and the humoral response, which we'll go on to talk about in more detail later on. So to defend the body from invasion by foreign material, lymphocytes must be able to distinguish between self and non-self, i.e. foreign material. If it couldn't do this, then lymphocytes would destroy the body's own tissues. So how does the immune system do this? Each type of cell has specific molecules on its surface that identify it. However, it is usually by the proteins on the surface by which the immune system can recognize if the cell is self or foreign. This is because proteins can be of many different types and have a highly specific tertiary structure. It is these molecules which allow the immune system to identify pathogens, cells from other organisms of the same species, abnormal body cells such as cancer cells or cells infected by pathogens which have antigens presented on their surface, as well as toxins. All of these are potentially harmful, which is why being able to identify them as foreign or abnormal is so important. It is often by the proteins on the surface of foreign cells or pathogens that the immune system recognizes them as foreign, and therefore it is often these proteins which act as antigens. The spec wants us to learn the definition for antigen, so an antigen is a molecule that triggers an immune response by lymphocytes. An antigen can be any part of an organism, such as a protein on its surface, or substance that is recognized as foreign by the immune system and hereby stimulates an immune response. Great, so now that we've given an introduction to immunity as well as antigens and their definition, we'll next move on to phagocytosis and the two types of specific immune response. So what actually is a phagocyte? Well, a phagocyte is a type of white blood cell which carries out phagocytosis. Phagocytosis being the mechanism by which cells engulf particles to form a vesicle or vacuole. In our scenario, this refers to the engulfment of pathogens. So how do phagocytes actually destroy pathogenic material? First of all, the phagocyte is attracted to the pathogen by the chemical product it has left behind. Then, the phagocyte attaches to chemicals on the surface of the pathogen via specific receptors on its cell surface membrane. Then the cytoplasm of the phagocyte moves around the pathogen, hereby engulfing it. The pathogen is now contained in a phagosome, which is a type of vesicle. Next, a lysosome containing lysozymes fuses with the phagosome, releasing its lysozymes into the phagosome. These lysozymes then hydrolyze the pathogen. The hydrolysis products are then absorbed into the cytoplasm and other debris is expelled by exocytosis. Next, the phagocyte presents the pathogen's antigens on its cell surface membrane. Hereby it activates other immune system cells. The phagocyte in itself is harmless but hereby activates an immune response. Note that the body also has specific responses that react to specific antigens. These are slower at first, but provide long-term immunity. The specific immune response depends on lymphocytes, which are a type of white blood cell. There are two different types of lymphocytes, T and B lymphocytes. Both originate in the bone marrow, but they mature in different places. The T lymphocytes mature in the thymus gland, hence the name T, and B lymphocytes mature in the bone marrow, hence the name B. T lymphocytes are associated with the cellular response, also known as the cell-mediated response, and B lymphocytes are associated with the humoral response. When comparing the two types of immune response, the cellular immune response is immunity involving body cells that have antigens presented on their surface. The humoral response, on the other hand, involves antibodies that are present in body fluids such as the blood plasma. So first of all, we have the cellular response. This involves T lymphocytes, T helper cells, binding to antigens presented by antigen-presenting cells, also known as APCs, which are cells that present antigens of other cells on their own cell surface membranes. Antigen-presenting cells include phagocytes, which have displayed antigens of foreign cells or pathogens on their surface after phagocytosis, body cells which have been invaded by a virus and have presented viral antigens on their surface. They also include cancer cells which present antigens on their cell surface membranes and transplanted cells from other individuals of the same species. So how does cell-mediated immunity work? First of all, a pathogen invades a body cell or is taken in by phagocytosis. Next, the invaded cell, or the phagocyte, presents the antigens of the pathogen on its cell surface membrane. They are APCs. Next, a T helper cell binds to the antigen via a specific T cell receptor. This activates the T helper cell to divide rapidly by mitosis and form clones of this specific T helper cell. This is known as clonal selection. These cloned T helper cells then may develop into memory cells that enable a rapid response to future infections by the same pathogen. They may also stimulate phagocytes to engulf pathogens by phagocytosis, or stimulate B cells to divide and secrete antibodies in the humoral response. They may also activate cytotoxic T cells. So what do cytotoxic T cells actually do? Cytotoxic T cells kill abnormal cells and body cells that have been infected by pathogens. They produce perforin, which is a type of protein. This perforin makes holes in the cell surface membranes of cells, which makes them fully permeable to all substances, and the cell dies. Note that cytotoxic T cells are most effective against viruses, as viruses hijack body cells to reproduce, and therefore killing host cells prevents viruses from replicating and going on to infect more cells. Nice, so now that we've covered phagocytosis and the cell-mediated response, we can move on to the humoral response. So the humoral response involves antibodies that are soluble in the blood and tissue fluid. So the spec requires us to know the definition of antibodies. So here it is. Antibodies are proteins produced by B lymphocytes in response to the presence of the appropriate antigen. We also need to know the structure of antibodies, so make sure you know this diagram. In an exam, it is often helpful to draw this diagram, and you will be credited for this. An antibody is a protein with a quaternary structure, meaning that it consists of a number of polypeptide chains, in this case 4, joined together. To recap proteins, just click on the button bottom left. The 4 polypeptide chains that make up antibodies are linked by disulfide bridges. There are 2 heavy chains and 2 light chains. Each chain consists of a constant region marked in brown, which is the same for all antibodies, and a variable region marked in green, which is different for different antibodies. Each antibody has 2 identical antigen binding sites, which are complementary to one specific antigen. Antibodies bind to antigens on the surface of pathogens to form antigen-antibody complexes. Note that the massive variety of antibodies is only possible due to the variable tertiary structure of proteins, as antibodies, as I mentioned, are proteins. Each B lymphocyte produces a different type of antibody which has antigen binding sites specific to one antigen. When the body is infected by foreign material, the B cell that produces antibodies specific to that antigen is stimulated to divide. So how does this actually happen? First of all, surface antigens of an invading pathogen are taken in by a B cell by endocytosis. Next, this B cell processes these antigens and presents them on its surface. T helper cells, which were activated in the cell-mediated response, attach to the processed antigens on the B cell, hereby activating it. This B cell then divides by mitosis to give a clone of B cells. This process is called clonal selection, whereby the B cell specific to that antigen is activated to divide. The clone of B cells are called plasma cells. The plasma cells produce and secrete antibodies specific to the antigen on the pathogen's surface. The antibodies secreted by the plasma cells bind to antigens on the surface of the pathogens, causing them to agglutinate, making it easier for phagocytes to locate and engulf them. They then serve as markers that stimulate phagocytes to engulf the pathogen. Some of the B cells develop into memory cells. These can divide rapidly and develop into plasma cells if the body is infected by the same pathogen again. This is called the secondary immune response. Again, just going back to clonal selection, remember this term as it is often credited in responses in exams. So just a quick reminder, clonal selection is when the specific B cell to that antigen is stimulated to divide to build up its numbers to a level where it can be effective in destroying this pathogen. So just to avoid confusion, plasma cells secrete antibodies usually in the blood plasma. They're involved in the primary immune response, which is a slower response, and memory cells circulate in the blood and tissue fluid. They're involved in the secondary immune response, and this is a rapid response, so a person will not get ill. When comparing the primary and secondary immune responses, you may get shown a graph looking a bit like this. And you can see that the secondary immune response, which is after this dotted green line here, has a higher concentration of antibodies produced at a faster rate. There's a shorter lag time, the antibody concentration peaks higher, and also the concentration of antibodies remains higher for longer. And this is because memory B cells from the first exposure to the antigen can divide rapidly into plasma cells that produce the specific antibody to that antigen. So in effect, clonal selection happens faster, also due to the presence of memory T cells. So overall, a high concentration of antibodies is produced at a faster rate, which does not allow the pathogen to replicate enough to produce symptoms. Next, we'll have a look at vaccination. Vaccination is the introduction of small amounts of antigens or dead or inactive pathogenic material to stimulate an immune response against a particular disease. Vaccines stimulate the production of memory cells against a particular disease without causing any symptoms. If the body is exposed to the same antigen again, antibodies are produced rapidly and the infection is overcome without any symptoms. If a large proportion of the population is immune to a disease, for example through a mass vaccination program, then the spread of that pathogen is greatly reduced, because it is less likely that a person will become infected and pass it on. This is known as herd immunity. Herd immunity means that vulnerable members of the population are protected and the disease may disappear altogether. Note, however, that antigenic variability may mean that a vaccine will no longer be effective against a particular disease. This may be because a pathogen mutates to have a different antigen, so it no longer is recognized by the immune system. Next, we'll have a look at active and passive immunity. Active immunity is immunity resulting from the stimulation of an individual's own immune system through exposure to the antigen, i.e. B cells produce antibodies, and passive immunity is immunity resulting from the introduction of antibodies into individuals from an outside source. So I've just put it all into a table to make it a bit more visual. Natural active immunity is infection and recovery. Natural passive immunity is, for example, antibodies received from the mother via the breast milk or placenta. Artificial active immunity is vaccination, i.e. the antigens are introduced by a jab. And artificial passive immunity, for example, could be the injection of antiserum containing antibodies after you've been bitten by a snake. So just to compare active and passive immunity, active immunity requires the exposure to the antigen. However, it takes time for protection to develop, whereas in passive immunity, you don't require exposure to the antigen and we have immediate protection. And in exams, they want you to write passive immunity is faster. In active immunity, memory cells are produced, and because of this, we have long-term protection due to these memory B and T cells, which stay in the blood for years. Whereas in passive immunity, no memory cells are produced, and we only have short-term protection as the antibodies are broken down shortly after. Great, so that would be the humoral response, antibodies, vaccines, and active and passive immunity. That's the end to part 1 of cell recognition and the immune system. To watch part 2, just click on the video bottom left.